So I want to focus this particular presentation on fungal genomics because my understanding is that this is okay. First of all, do you hear me? Everything's fine. Do you see my screen?
Yep, looks good.
Okay, good. On fungal genomics, because it feels like it's a little bit of underserved community, and there are some high priority pathogens there. So I'm gonna explain what we have in BRC analytics. I will explain what BRC analytics is, and then I'm going to give you an example of RNA-seq analysis. That's more or less the outline. It's all a bit of a bleeding edge, I would say, because I'm going to use some crazy agentic AI tools and things like that. Oh, whatever we'll be able to fit in half an hour. So in the past month or so, we ingested a lot of fungal genomes. So if you actually go to BRC Analytics website, which is brcanalytics.org, if you scroll here, you will see this phylogenetic viewer. If you click on fungi, so you will see so we have about 3,000 assemblies. Now we have organisms, but we have assemblies. So there's sort of two categories. So organism, that's a species, but species can have multiple versions of the genome associated with that species. So that's what assemblies are. For example, you can have multiple strains, things like that. So there are more assemblies than species because there could be multiple assemblies per species. Now, the species I want to play with today is Candidazima auris. So let's find the data which we have for that pathogen. And again, in order to do this, you go to BRC Analytics, you click on organisms, and you just start typing. So here it is. And you can see that there's the species. We have 6 assemblies associated with this. Now, I don't know how this looks on your screens. It could be very small. So maybe we'll increase this a little bit. So these are the assemblies associated with this particular species. So there's some information about these assemblies, essentially quality and so on. This is I guess it's a bit of so for me, I'm not a fungal person. So for me, I would probably go for the best assembly if I were doing the analysis of Candida auris. And best assembly for me would have high and 50 values. But that's not necessarily what the people who actually know this species will do. And for example, one of the strains, so majority of studies that involve some genomic data for this pathogen, they work on B8441 strain. And so this is the assembly we're going to play with today. Now, what does it mean play? So in this particular webinar, I wanted to look at data presented in this paper and show you how it can be replicated. This is a science paper which describes SCF1, which promotes adhesion in Candida auris, why it sticks to things. This is also a good study because it's kind of a straightforward RNA-seq. Most of the studies, most of the data that you can find for Candida auris, so there are really 2 types of data sets for Candida auris in NCBI or on short-read archive. That's the whole genome data sets generated by state health agencies. There's a lot of data going in that sense. Plus, there's a lot of RNA-seq data sets. RNA-seq is interesting because it's, I guess, one of the less straightforward analyses for the simple reason is that it's you can kind of separate this into 2 groups. I'm talking about RNA-seq designed for differential gene expression, because it kind of consists of 2 steps. First, you just generate the counts. So you have samples, you align them against the genome, you align the reads against the genome, find reads associated with genes, and count how many reads I have per gene. So you do it for those samples. But then comes the actual differential expression analysis. And for that, you need to explain to the tool that you're using how your data is structured.
Sense today. Congressman Barnes, wow.
Yeah.
I never really thought.
What are the conditions for each condition? What are the replicates? Essentially, what are you contrasting? So this is why, so it's a good example. So how would I replicate this analysis in, say, BRC analytics? Why I'm replicating this? Because, of course, in this case, I know what the final result is. So it's also a good way to validate that what I'm doing is actually correct. But what I'm going to present to you here is it's a great way to do generic differential expression RNA-seq via BRC analytics. So what I would do is papers like this, they usually specify how they deposited the data. So for this paper, data is deposited in CBI, everything is good. It has a project ID by a project ID, which is listed in the paper. I just have it here so I don't forget what it is. So I want to replicate this via BRC analytics. So I already found the genome. This paper did use B8441, but there's an interesting twist to this. The paper is from 2023, and so the paper used actually a different version. So it used, it's the same number, but the version is 0.2 in the paper. Actually, I don't know which version of the paper, I don't remember where it states it's explicitly or not, but that is going to be a problem many of you will face. How in the hell do I compare gene IDs from this assembly to gene IDs from that assembly? That's actually a very hard problem sometimes. And the take-home message here for those of you who are listening to me and actually generating the data, please, when you submit, when you write your materials and method to supplementary data, please state that clearly. It's going to help, well, I'm sharing the wrong tab. It's going to help you, it's going to help everybody who's trying to replicate your data. Metadata is important for a reason. So I think I forgot to share the right screen. What I'm saying is that now I'm in BRC analytics. We already found Candida zima auris. We're looking at the assemblies. I'm going to choose this one. But what I was mentioning is that this is the latest assembly, but the paper probably used the previous one. So now I can go ahead and click Analyze. And this would give me a few options. Sometimes I just want to grab the data from BRC analytics and analyze it myself. I don't, you know, I have some ideas how I want to do this. Or we have a collection of workflows. This collection is going to be expanding. It's built on community annotated workflows. And so here are your options: variant calling, transcriptomics, and so on. We are still in the process of getting this in front of the people. So we will be improving kind of a hand-holding. We'll be improving descriptions, exactly what this is, providing more help, and so on and so forth. But so for transcriptomics, we have quite a few. You can do single-cell RNA-seq with this platform, but this is not what we're doing here. Here we're just going to do straightforward RNA-seq for paired end data, because this paper has a typical Illumina paired end data sets. And so I'm going to choose this workflow, and I'm going to click Configure Inputs. So this will bring me to this screen. What is RNA-seq analysis in this case? I have the reads that I generated in the lab. Well, in this case, I grabbed them from the paper. I need to align them against the genome. I map them against the genome. I already know which genome that would be. When I'm doing RNA-seq analysis of this type, I'm generating counts associated with genes. So whatever workflow I'm going to use needs to know where the genes are. And so there are several so-called tracks, or you can also, because you can have official and CBI genes, you can have gene predictions, you can have community-supported genes, things like that. So in this case, I'm going to use NCBI gene. I'm going to click Continue, and this is where you provide your data. So here you can either provide your data that you generated in the lab, or in this case, I will be grabbing stuff from NCBI. This interface automatically looks up which data already exists in SRA, so that about 26,000 IDs. But in this case, I know what I'm looking for. I'm looking for this data set. So I'm just gonna instead click enter accessions and just, well, paste this thing in here and click continue. So it will look up this bioproject ID. For those of you who are not familiar with bioproject ID, it's a kind of a identifier for a group of data sets associated with a particular study. So in this particular study, there are 6 data sets. And it tells you their accessions, all the information about, this is paired end data, so you're going to have 2 files associated with it, and a lot of other information. So we can select them all, click add sequencing runs, and at this point we can go ahead and actually launch this workflow. So this will bring you into Galaxy, which is a platform which actually does the dirty work of actually running the analysis behind the scenes. So what this will do, it will open a new tab. Let me make this a little bit bigger. And it will be pre-filled already for you. So the reference genome, it's pulled from BRC Analytics. It will specify the data for genes. We also selected the genes. It also already pre-populated with these paired end reads, which we chose by entering bioproject ID. This is not completely automatic transmission. There is a little bit of manual action here. And that manual action is that you kind of need to know what kind of RNA-seq library was you used. When you're doing your own experiments, you probably know that. So whether it's stranded or unstranded, in this particular case, it's actually stranded reverse. But this is yet another example. When you guys write material and method sections for your papers, don't just mention the kit. Mention how library was prepared. This is also very important. It's very hard because there is no structured way of providing this information, and SRA metadata doesn't list that. This is particularly important in the age of LLMs and all this AI stuff, because that AI stuff pulls from that metadata. So in a sense, by describing your experiments that you publish as much as possible, you're actually promoting your research, because it will be easier to find it. It will be easier to promote it. It will be easier to understand what you did. So again, please, please, please tell your students, when they fill those boring NCBI forms, they're doing this for a very good reason. And unfortunately, it's hard to make them more entertaining. And please, applaud to all the work that NCBI is doing. Without them, none of this is possible. So, and then you would click run workflow. Since we only have 15 minutes left, I'm not going to do this. I'm just going to, of course, I pre-ran it. And when you start running this, it looks something like this. So you actually get the workflow. Well, it looks something like this. You actually get the workflow so-called invocation. And so the green boxes here represent how this sort of went. There is a bit of a simpler representation of this workflow. So actually what's happening here is you get the paired end data, do some QC, then you use star mapper. It's a spliced RNA-seq mapper, which maps the reads, and it also does the counting for you. But this workflow contains kind of so many boxes compared with this representation, simply because there are lots of options, because you can configure it to use feature counts, or you can use strings. So there are many possibilities here. I think that the fast P to star to gene counts is straightforward and quite sufficient here. Sometimes simple is better. So so you get something like this, and at the end of the day, it's done. What does it mean? It means that you have, this is, again, if you're not used to Galaxy, then that might feel a little bit overwhelming, but you get your data in. So each of the steps in this workflow generates a data set. This is it's like your workspace. It's like a folder associated with this analysis. And these are the kind of a final data set. So what this workflow really does, it generates counts. And these counts, they look like this. So they look like so gene ID, how many reads are associated with this particular gene. I think I'm showing you the wrong history here, so let me correct that. Yes, this is the, sorry, 1 second. Okay, yeah, this is the right one. So the correct counts there, because we have 6 samples, so we have these things. So now the hard part starts. So I have these 6 samples, right? So now the paper that I was showing you, and so this paper, was actually doing 3 kinds of comparisons. So there was a wild strain, which was compared against a mutant and another strain. So I have 2 types of contrasts here. I'm comparing wild type against one treatment, let's call it treatment, and I'm comparing my wild type against another treatment. So I need somehow to figure out what is what. And again, if you're doing this experiment, you probably know that. But if you are reusing somebody's data, like in the case of this paper, then you need to do a little bit of forensics. Sometimes, like in case of this paper, the supplementary data is really good, and you can actually understand what is what. Sometimes you can just download data from NCB, from SRA, and SRA metadata is good enough to provide it. So this is kind of a rare situation. So it's very difficult to predict for different publications how this is what. So the way I did this in particular, I used agentic AI. So I used kind of something that is powered by LLMs. I did not use ChatGPT. I did not use Claude in the clear sense. I didn't go to web interface. I used something which is called Claude Code, and there's a similar tool from Google called Gemini Code Assistant. For those of you, again, who are from a fungal community, you might feel like, oh God, this is yet another thing I need to learn. Since this webinar series is kind of called Tools of the Future, then this is where we're going. So the Claude Code in this case integrates with Galaxy. So you can essentially orchestrate things in Galaxy from Claude Code. Again, it's a half-an-hour webinar, but we're going to have a white paper describing precisely this analysis published. It will be on BRC site sometime next week, so you can actually in details read in details what I'm talking about. But these kinds of investigative actions, given the paper and its supplementary data and the SRA IDs, figure out what is what, are kind of perfect for this kind, for agentic AI. So essentially what I did is I downloaded all the Excel, all the metadata from the paper. I downloaded paper itself. I downloaded, well, actually, this is all I did. And then I put it in a folder, and I just asked the agentic tool, so given this PDF file with this supplementary data, look at this particular count table with these IDs and tell me what is what. The note here is that you can never trust anything that AI gives you. You need to validate, but at least it provides you this report in a structured way that it's easy to validate. So what happened in the end is that it partitioned my data in in data sets based on, so it's actually figured out what is what. It figured out what is AR382 wild type, what is AR0387 wild type, and what is the mutant. So it essentially restructured my single set of data sets of everything pulled together into 3 separate entities and told me what were the comparisons in the paper. I don't trust it, so I go to the paper. I've actually verified that it's in fact the case. And it was the case. And at that point, you can go ahead and run DE-seq2. In this case, I essentially ran DE-seq2 in Galaxy directly. So this is DE-seq2 interface. You say, this is my, so this is one factor, this is one condition, this is another condition. Go ahead and perform differential expression analysis, which it did, and it generates, typical DE-seq output. And at this point, I mean, it depends what you're doing. So if you are doing this in your own research, or it depends what your experimental design is and what you want to do with this data. In my case, I wanted to compare it with the results of the paper because I wanted to know that what I'm doing is more or less correct. And I again employed the same Claude Code idea. Essentially, I gave it these data sets and I asked, okay, given this, take basically supplementary data from the paper, because supplementary data in the paper listed, they had a similar outputs of DE-seq, and tell me, do they kind of look similar to each other? Am I reproducing them, am I replicating them correctly? And then it hit the classical problem. Gene IDs don't match because paper used 0.2 version and I used 0.3 version. But again, the beauty of this kind of agentic AI, when you're using this properly, when you have a folder where you put all the data, this is not something you want to do from Claude or ChatGPT interface because it's going to get lost in this. This is not something you want to do in the classical way as we used to use chatbots. And I basically told it, all right, so we have several options how we can figure this out. So basically grab previous version from NCBI and check gene descriptions. And if this doesn't work, grab the version from NCBI and check protein sequences if they match. And it in fact did that. Where was this? And so at the end of the day, it showed me, it performed essentially the correlation analysis between the two. Let me find the right slide here. And these correlations were quite, so in fact, what my workflow was doing was very much, let me magnify this a little bit. So this is the wrong paper because I did validation of 2 papers.
Anton, I don't think you're showing us the right screen.
Thank you. So this is correlation between my results and results which were in the paper. Almost too good to be true. But then, how do I make this reproducible? How do I give these results to somebody else? How do I share this? How do I generalize this? Because I can type whatever I want in these agentic AI tools, but how do I share these artifacts? Well, what we did next is we produced a Jupyter notebook, which doesn't need any AI. It essentially replicates this analysis. So, and this notebook is in Galaxy. It has access, so I can use this notebook to essentially tell it which data sets were produced by DE-seq2 by me, and I can download the data, the differential expression results from the paper, and I can rerun the whole thing, and here are the same plots. but in this case, in a kind of a much more interactive way. So, I'm sharing again the wrong tab. So what I did is I went back to Galaxy. There's a notebook in my history which I can start. This is Jupyter Light Notebook. It runs in the browser directly, and here is this plot. So ultimately, these two approaches, using Galaxy for structured analysis, for running the workflows, and for keeping everything on track, and this kind of a non-linear LLM agentic AI type of stuff should be integrated into one. This is our next. This is what we're working on. But for better or worse, the future is in this. And I guess take-home messages here are: first, I really would like the fungal community to start using this and giving us feedback. You can give us feedback by using this question mark on BRC Analytics website. You can, because one of the things we want to know, does it work for you? Which other genome assemblies you want to add? And another take-home message here is that never trust AI. Always validate and doubt it. And in fact, the way we approach this problem, we actually don't ask AI to do things. We ask AI to generate a plan for what it thinks needs to be done, and then we review this plan. So it's not completely crazy. And finally, it's this combination between this flexibility of LLMs and rigidity of Galaxy keeps the provenance and keeps things reproducible. And my fourth point here is that please tell your students to record metadata correctly, and please record which versions of the genomes you use in your applications, because otherwise it's basically wasted effort if it cannot be reproduced. I think we have 30 seconds for questions.
Thanks, Anton. If anyone has questions, feel free. But Christoph, if you want to talk about next month, what we have coming.
Yeah, well, thanks, Anton. That was really fascinating. I also just want to say hello. I'm from SIB on the PDN. I look forward to meeting many of you. I just want to mention before we finish that the next Tools for Tomorrow will be on the 9th of January. And we have Anya Parker from our side who will talk about Localus and how it's used also in the Pathoplexus data repository. So you'll join in to hear about this, and if there's some more questions, maybe some people can still ask Anton, but I will have to leave now. But thank you very much for the nice presentation.
Thank you.
And across all BRCs, we have a YouTube channel that these Tools for Tomorrow webinars will be posted on, and we also have the hackathon that the BRCs participated in last month. Videos from those are on there now. So I put that link in the chat. Feel free to check into it, and I'll let Rebecca ask her question.
I was just curious, those data sets were large. How long did that whole thing take to run?
So you mean RNA-seq analysis?
Yes.
15 minutes, half an hour.
Nice.
So this is actually an important question, because I think I want to show you just for 1 second this thing here. Just give me 1 minute. I want to show you this. Oh no, this is the wrong slide. This one slide. It's very complicated. This is more complicated than RNA-seq for sure. So BRC is built on the Access CI infrastructure, specifically on Texas Advanced Computing Center infrastructure. So there is some beefy infrastructure behind that. but these are small data sets, relatively speaking. It's only six accessions. We're also doing reanalysis of the population sequencing data. These are about 20,000 data sets. That, of course, takes a little bit longer. But most of the analysis, at least in fungal space, are not